This is the final part of olfaction and the final part of the chemical senses. We're going to bring olfaction and taste together so that we can experience flavor after discussing higher order processing in olfaction. So as usual, we want to talk about the higher order olfactory processing, or what's going on when we get to the cortex. And I'm going to start by this talking about olfaction in the environment, and I'm going to talk again about this idea of how do we perceptually group specific molecules together for smelling a particular odorant. How do we segregate all the molecules into their various components to go with the correct odorant. And it seems particularly complicated for olfaction, especially we rarely smell one chemical in isolation. So he gives us the example of coffee, which has more than 100 different molecules. Plus, a lot of times when we're smelling coffee, we can smell coffee even when other odorants are present, even when other smells are around. So if I come downstairs to the kitchen and there's orange juice there and bacon and the smell of coffee, I'm smelling all of those different smells, and yet it doesn't, all of those molecules are in the air, and yet instead of smelling some weird bacony coffee thing or something where those molecules get segregated differently, I'm smelling coffee at times, and then other times I'll be smelling bacon will seem like the stronger smell, the smell that I'm paying attention to. And how do we do this? How do we perform this kind of perceptual organization for olfaction? And you can see my green questions here. And then I also remind you of vision. If you remember talking about, we did this for vision, and we had a whole practice in class discussing this for hearing, how we might use things like similarity. I'll use the hearing of similarity of timbre and pitch. We're going to group that together into a melody, or group that together in maybe into a particular instrument, as opposed to if they differ in timbre and pitch, we're going to segregate those into different instruments. We might use good continuation, proximity, common fate, synchrony, meaningfulness, familiarity. So those are just some reminders of what we've talked about for other sensory modalities. And then I ask, what do you think? Anything else? What are we doing for olfaction? Again, I'm not smelling a bunch of molecules from the coffee as mixing with some of the molecules from the bacon. And vice versa, and other molecules mixing with different molecules, such that I go, what is that smell, right? Somehow I'm smelling coffee and bacon. I keep leaving out the orange juice because it seems like a smell that travels less, but if I put my nose up to the orange juice, I can smell the orange juice as well. And I do want to pause here. I do want you to think about that. I want you to pause this and think about that before I give some answers that students have given in the past. There is no, here's the simple answer. I'm gonna say I think that a lot more of this is top-down processing than what we've seen before. That the bottom-up processing doesn't really explain things for me, although there might be a kind of proximity if there is some segregation to where those molecules are in the air, and that they are closer to each other when they are from all the molecules from coffee, let's say, are going to be closer to each other or closer to the coffee, and that maybe we can walk through bits and pieces where we have some bottom-up processing, but to a great extent this seems to be we need to have the experience of smelling a particular smell before we can really segregate that from the other smells that are around it. And so I'm going to say top-down processing, but I don't think that's the whole answer. And I don't think there's an easy answer. I don't know how much people have examined this the way they have examined vision and hearing. So it's a fun thing to think about. So I have this picture again of the olfactory mucosa. That's where our olfactory receptor neurons are, the sensory neurons. and that those sensory receptors are communicating to the olfactory bulb, which is immediately communicating to primary olfactory receiving area, the piriform cortex, and the amygdala. There's lots of crosstalk here between those areas and the olfactory bulb. There's crosstalk here between those areas and each other, and there's crosstalk between those areas and the orbitofrontal cortex. And when we're going to talk about now higher order olfactory processing, remember this is getting information to the cortex, and really we're going to include the piriform cortex here, but I'm going to get much more specific about what's happening with that orbitofrontal cortex. And you can tell from the picture of the brain there. So this is named after, we have the prefrontal, it's part of the prefrontal cortex, and we have that kind of ventromedial prefrontal cortex in the middle there, and the orbitofrontal cortex is really kind of around the orbits of the eyes, if that helps you to remember that. It's just kind of right up above the orbits of the eyes, thus its name, the orbitofrontal cortex. So in the piriform cortex, odorants cause widespread activity here that can change via experience. If we go back to the olfactory bulb, that is a chemotopic map that is based on the chemicals themselves, the chain length, and our perception, what that odorant is going to smell like. And that's not going to change no matter how much we smell that smell, or whether it's the first time we've ever smelled that smell. That activity of the olfactory bulb is going to be the activity, just like visual information is going to be the visual information that we experience. But as we get to the piriform cortex, this idea that odorants cause widespread activity, that is what I was explaining before with the picture where we went into the piriform cortex, and I mentioned that it's not just communicating to the amygdala and the orbitofrontal cortex, it is communicating across the hemisphere to the entorhinal cortex, to the perirhinal cortex, to the limbic system, to so the amygdala and the orbitofrontal cortex as well. So all across that hemisphere, those neurons are communicating to many different areas, to thousands of other neurons, and that's widespread activity. And that activity is going to change based on experience. Goldstein gives us this nice picture of, let's say, so I have this odor object like a rose. I smell it. Every time I smell it, I'm going to have that same activity on the olfactory bulb, as this is just some example of where the olfactory bulb might be active. And then when I go to the piriform cortex, I'm going to see this scattered activation. Then as I smell that object, that odor object more and more, we see the connections between the neurons are going to, the pattern of activation can change and the connections between the neurons can change. And this is just showing within the piriform cortex, let's say, those connections that are built based on my experience, but that's going to be connections, again, throughout the hemisphere. And so to some extent, this is going to be how it is going to change based on just how well do I know the odor object. So I usually do a smell lab or a smell demonstration, and one of those odor objects is coffee, and most students recognize that very easily. We've had lots of experience with coffee. That's going to change that activity in the piriform cortex, that you're getting more and more experience with this. But there's also going to be changes depending on your specific experience. So did you have a really good experience with smelling coffee? Have you had a really bad experience with smelling coffee? That's going to change those connections as well. And this Donald Wilson study, given time, we can discriminate between odorants. He did a study with rats where they gave them either isoamyl acetate, if I'm remembering that correctly, it's a banana type odor, and they gave them another odor object of the isoamyl acetate mixed with another smell. I want to say pineapple, but I think I'm mixing, a peppermint smell. And the rats were either allowed to sniff the odor object for 10 seconds, so about 20 sniffs, or they were allowed to sniff those odor objects for about 50 seconds, so about 100 sniffs. And if we look at what's happening in with their experience and with the pattern of activation for those odor objects in the piriform cortex, later on we did not see much difference between the two smells for the conditions where the rats could only smell that for about 10 seconds, as they were not getting enough experience to really distinguish those. But if they were given 50 seconds to smell each of those smells, they were able to distinguish those. There was a difference for them in their perception of the two odor objects, and they could tell the difference. And we see a change in the pattern of activation in the piriform cortex. When we get out to the orbitofrontal cortex, that is now assigning an affective value to stimuli and experiences. And so that's, if you remember, that communication is going both ways, and it's talking back, communicating back to the amygdala and to the piriform cortex, which is why I said whether or not we've had good or bad experiences can influence that activity in the piriform cortex, as that orbitofrontal cortex assigns an affective value. In this study, they gave people, this is a little bit gross if you think about it, but they gave people a mixture of isovaleric acid. So this is something that smells a bit like sweat, and a cheddar cheese flavoring. And if they were told this is cheddar cheese, people rated that as more pleasant than if they were told this is body odor. They rated that as less pleasant. Again, it just doesn't sound very pleasant at all. But apparently, if we're told, if it has that cheddar cheese flavoring as part of this, as part of the odor, and we're told it's cheddar cheese, maybe we don't notice the isovaleric acid, this kind of smell, sweat smell, so much. And then if we're told it's body odor, obviously we do, and that cheddar cheese flavoring is probably not a great smell to have in there, and so rated as much less pleasant. And we see differences in the orbitofrontal cortex. I think I actually have this on a slide here. I'm going to go ahead and move while I'm talking this time. Yes, where if we look at fMRI activation, this is a percent change, right? They have a control condition that they subtract out of this, out of examining the cheddar cheese or the body odor. And you can see that if we compare this to a control condition, and I'm told that it's cheddar cheese, I'm rating this as pleasant, and I have activation in the orbitofrontal cortex, okay, excitatory activation in the orbitofrontal cortex. If I'm told this is body odor, I not only rate this as unpleasant, but it almost looks like I'm inhibiting activity in my orbitofrontal cortex, as we are below my control condition in this case. I think that's interesting. We're actually just kind of doing this, don't like it, and we're inhibiting activity there. Now, as we're at this orbitofrontal cortex, we can talk about flavor, where we're going to put together taste and olfaction. So again, flavor is really taste plus olfaction, those things coming together to give us the perception of flavor, for us to have the perception of really the overall dining experience of our food. As I think I say maybe one more time here, you know, we are also taking into account the feel of the food, the looks of the food, and one of my students did a project showing that the sound of the food is also important. But we're going to start with this olfaction as being part of flavor, in addition to putting it together with those basic taste qualities. You might want to do the demo on page 378. My memory is it's this kind of, you're tasting several things, and then you pinch your nose as you're tasting those things, and you'll notice how much less taste or flavor there really is when you take your olfaction out of it. And of course, this has become something they are talking about more and more in the news, as losing taste and olfaction is something that can happen to people who have the coronavirus, and they're especially more likely to lose some of that. Those olfactory receptor neurons are blocked for a little while, and it affects flavor so much. But what's happening is, as we are putting that solid or liquid into our mouth, and those chemicals are being taken in by those taste receptors, that the volatile chemicals are also going up through this nasal pharynx. Okay, so through the back of the mouth and up into the nasal cavity, and then hitting the olfactory mucosa in that way. Your author includes here some data from a study looking at a few different chemicals, and then so open in this example is people leaving their nose unpinched, so their olfactory receptor neurons, they're open basically for receiving the odorant, versus clamped. Okay, I'm going to go through these. The sodium oleate, which has a kind of soapy taste, as their
olfactory receptor neurons are open and the nose is open and they can smell that. People rate that. Most people are rating that as soapy. You can see there's just a couple X's in other places. It's just certain individuals taste that a little bit differently. If people clamp their nose, most people are saying that that is tasteless. If we look at ferrous sulfate, most people are saying when they can smell it that it is a metallic kind of taste. And again, if they clamp their nose, a lot of people are saying this is tasteless. Here there's a little bit more individual variation there. MSG, which we have talked about as an umami taste, in this example, when the nose is open, people are rating that as salty. When they clamp their nose, they're still tasting that as salty. So MSG is going to have the same flavor whether the nose is clamped or not. So many sensations that we are calling taste are really greatly influenced by the stimulation of the olfactory receptor neurons. And there are some, though, that are more influenced by those basic taste qualities.
So a number of cortical areas that serve both taste and olfaction are probably involved in our physiology of flavor and how we experience flavor. But most of the research has been done on the orbitofrontal cortex, where we really see all of these different sensory modalities coming together so that we have this experience of not just flavor, but of our food, of the experience of eating our food. This is where taste and smell responses are first combined into something more complicated like flavor. But the orbitofrontal cortex also receives input from the primary somatosensory cortex, so the feel of our food, and the infratemporal cortex, so the looks of our food. The infratemporal cortex in that visual what pathway, so where we do our object recognition. If you've ever done the green eggs and ham when you were a little kid, my sister does that with her 4-year-olds, usually every year. Guess they're probably not doing that this year, but and as the kids are rating, she also collects descriptive data and shows them their descriptive data of who liked the green eggs and ham versus not, compared to regular eggs and ham. Some children don't like the green eggs and ham, even though it's exactly the same as the regular eggs and ham that they eat. That information, those eggs being green and that ham being green, just that influences their experience of the food. And certainly how food feels in our mouth is very important as well. But the orbitofrontal cortex does not just receive input from the primary cortical areas for taste and smell, so the insula, the frontal operculum, the piriform cortex, the somatosensory cortex of the feeling of our body, of our mouths in this case, and the infratemporal cortex, so what that food looks like. But I have had students do their project on the importance of the sound of the food, so the sound of popcorn popping or the sound of our potato chips crunching. That also influences our experience of food. And the orbitofrontal cortex is putting all of that information together for flavor with taste and smell, but also for just the overall experience. I think he talks in the book a little bit about, I don't know if he took this out, about a particular soup that he has eaten that has the hints of lemongrass and the depth of mushrooms, and he has all of these flavors that are combining to make this really amazing experience of a soup. And the orbitofrontal cortex is doing this via bimodal neurons, so neurons that respond to more than one sense. So far, we've mostly talked about neurons that are responding to vision or neurons that are responding to hearing, and we have to put that information together. But we do have bimodal neurons, and they tend to respond to similar qualities in the environment or qualities that are going to tend to occur together in the environment. So for example, a cell that responds, a particular neuron that responds to the taste of sweet fruits would also respond to the smell of sweet fruits. And the orbitofrontal cortex is said to be the cortical center for detecting flavor and the perceptual representation of foods, as we are now experiencing several modalities for our experience of food. And we're putting them together with these bimodal neurons.
So I'm going to come back one last time to this relationship between olfaction and emotion, and just remind you how smell, that sense, does not go through the thalamus. And what the thalamus is doing is it is filtering, organizing, summarizing, and sending information on. We're skipping that step. We're going straight from the sensory neurons to the olfactory bulb. That olfactory bulb looks a little bit like other primary receiving areas, even though now we're just like up above the nose there. And then the piriform cortex, which is considered the primary receiving area, it is communicating directly to those memory, emotionally associated memory areas. It is communicating across the hemisphere. We also see communication to the amygdala, but then the pararhinal cortex, the interrhinal cortex, the hippocampus, and the orbitofrontal cortex, where we are putting that affective value onto things. This is really what causes this Proust effect, as it is called. The Proust effect really comes more from a literary background of Marcel Proust, talking about eating these madeleines and how they just transported him back to when he was something like 5 years old, and he can see the block that he lived on and things that he hadn't seen or experienced in years. And this is referring to the ability of taste and olfaction to unlock memories, and often these very kind of emotional memories or memories that are unlocked in a more vivid kind of way. So one of the things we see, and we're not going to talk about flashbulb memories at all pretty much in this class, but if you know your background in that, you know that one of the theories about flashbulb memories is that they are special because they are going through the amygdala. We have that emotionally associated learning, and so they do seem more vivid, but that they are not necessarily more accurate. And that's the same is true for these olfactory induced memories. They have more emotionality, they feel more vivid. It feels like we're remembering them more accurately and more completely, but they're not necessarily more accurate. We're still doing the same thing of pulling up a memory, talking about it. When I'm talking about my grandmother's sticky buns, I'm talking about it, and then I am biologically changing as I lay that back down. As we bring it up into that labile state, it is going to change when I am laying that back down. Okay, now this is my end of olfaction, so I'm hoping I've given you some. So if you read the connectivity of piriform cortex, if you read that journal article as an extra credit e-reserve, I'm hoping that you see the connections, and this makes it more, so that is a very technical article, and that this makes it more relatable to what you understand, to really experience.